Because it didn't make an embryo, it made a food tube. And there's your starfish. And here's your test for your starfish. It looks like no, not like that. It's exact, this is the picture. I know, because I printed it. And then I gave it to the lab specialist. I said, can you please laminate this for me? I'm not going to have them looking through the microscope. Because every time, they're like 2 years old. They got to touch things. And when they touch it, they move the pointer. And when they move the pointer, everybody behind, and that happened to me as an undergrad. And so that's like a pet peeve of mine. Don't make me get a question wrong, you butthead. So I avoid all of that. I put this thing out there. And you know what the other professor said? Yeah, you know what? I kind of like that too. So they use this. This is a picture that I printed off of my computer. Gary laminated it for me, God bless him. And now we use that. And that's what's out there. So what do you see? You see some gastrula. You see some morula. You see some blastula. That's what your professor's going to do. He's going to point to two of these things. Yeah.
Okay, so I see that one near the top is called a gastrula, but then the one on the top right is called a blastophore, and they look the same to me. A little bit high, a little
That's
bit.
the angle.
Oh, no. Oh, the angle.
This one here.
Yeah. No, the one to the a little bit lower to it.
This.
Yeah, that one, and then the top right one looks the same.
Way up there.
But it's called a blastophore, though.
No, the blastophore is actually the whole. That's the mouth.
Oh, okay.
So that's basically the opening of the tube.
Okay.
So they call him that the blastopore.
Oh, blastophore. Okay.
Yeah. The other thing that they kind of blew on this too, they labeled this thing a blastula. I'm sorry, that's not a blastula.
Okay.
That's a morula.
Huh.
That's not enough cells in there to be a blastula. So this obviously this is labeled, right? The one that's on your test won't be labeled. These are all blacked out. We don't go as far as larva. You will not be tested on the larva in the starfish. So you'll see the picture like this, but without all the labels on it. They'll all be blacked out. And they'll point to a blastula, a gastrula, or a morula. Because this one was labeled blastula, by the way, I never pointed to that on my test. I would not assume your professor's not going to do that. But that's not a blastula, that's a morula. I would argue, for a million years, I would argue with somebody about that. There's not enough cells in there for that to be a blastula. You got to be over 32 cells. That's blastula. Notice how blastula can look very different from each other, though. This is very early blastula. The cells are rather large still. Look at that. That's also a blastula. These cells in the beginning, by the way, they're dividing so fast they don't even have time to grow. They're getting smaller and smaller. Eventually they'll grow, but in the beginning, they actually get smaller and smaller. And so that's why you can have different presentations. That's a blastula, so is this. But so is this, and so is this. On my test, when I used to do starfish, I would use these as blastula. I wouldn't use those. Any one of these would be gastrula. And morula, I only even use this one or this one. This one technically is a morula, though. Okay, if I don't use this, what do? I put 4 questions on my human embryology. And so on human embryology, we got a morula. We still got that morula thing going on. And we got blastocyst instead of blastula. And that's because we're not going to make a gastrula. We're going to make an embryo. So on that purple poster board that you saw in your labs, there was a zygote on there. That's the single cell. All of these stages really basically are all morula. And then inside here, you got a very, very early blastocyst and a later blastocyst right there. And then there's the embryo. You could actually see the little critter in there, the embryo. But we're going to get more specific than that. This embryo, as it's forming, as this blastocyst is forming, you get this whole congregation of cells inside there, and that's called the inner cell mass. These, don't forget, this is dividing. These cells are dividing. Every second, boom, let's divide. Every second, boom, let's divide. So what we start doing, they start migrating into layers, and you're going to get 2 layers at first. And these layers then are going to form these empty spaces above it and below it. And very fortuitously, this yellow layer, what's that thing inside a chicken egg that's yellow?
Yolk.
Yolk. Yeah, that's it. We get yolk. You probably didn't know that. We actually make yolk. That's where our blood cells come from. Red blood cells, white blood cells, platelets, they all come from this yolk sac right there, this yolk cavity. Above it, you know this one too, because everybody in here has heard about that fluid that surrounds a baby inside mom's uterus. What is that called?
Amniotic fluid.
That's the amniotic cavity. There it is forming. Okay, now where is a yolk sac in the chicken egg? Right dead center in the middle, right? And so this layer of cells next to the yolk sac, that's going to be called endoderm, because endo means inside. This layer of cells that's forming next to the amniotic cavity, the amnion fluid goes around the outside of the baby. So what's our word for outside? Ecto. That's going to be the ectoderm. So we start with 2 layers of cells, ectoderm, endoderm, and then we're going to make a 3rd layer. It actually comes from the ectoderm. It makes like a volcano, but volcanoes have round holes instead of being round holes. Think of a long slit of a hole. And as these cells, remember these cells are dividing. As these cells divide, they climb up the volcano, and when they get to the top, they fall in. They fall into the volcano, and as they fall in, they create the mesoderm. So that's what's on my students' test. Endoderm, ectoderm, mesoderm, that is all together called the embryonic disc. You got the yolk sac below it, you got the amniotic cavity above it. There is one more thing that I like to test on, is where this embryonic disc is. At the edge here, one of these edges, there's going to be a stalk that starts to grow off. And the yolk sac is going to join that stalk. And you know it, the umbilical cord starts there. So it's called the body stalk in the beginning. It doesn't have blood vessels yet. Why? Because it doesn't have a circulatory system yet. That comes later, it will. But it's the body stalk that's forming. And we do have a name for that body stalk, and that's going to be called the allantois. So you got the amnion, yolk sac, and there it is, allantois. That is the body stalk. That's the body stalk. Cool stuff. This, by the way, is not a fetus yet. This is still embryo. Very late, but that's embryo. How come I can confidently say that? Because what is this forming here? You all know that.
Blood vessels?
Placenta. The placenta is forming. It's not a fetus until the placenta is done, because then you will have a fetal heart that's pumping blood through the blood vessels on the fetal side. Mother side, these overlap, they never connect, by the way. Fetal blood and maternal blood don't mix during the pregnancy at all. But the nutrients from maternal blood diffuse across the membranes and get into the fetal blood. And the garbage that's in the fetal blood diffuse out and gets into the maternal blood, because mom can take care of that. Mom uses her kidneys and liver to get rid of the garbage. But placenta is when you finally have fetus. So you got to have fetal heart first too. There's the yolk sac, see, isn't that cool? And notice how the blood vessels are coming from that, because it's filling that with blood. Okay. So in my class, I do 4 questions on this. All you other ones, your professor's probably going to do 2 questions on this. Not probably, they will. They'll do 2 questions on this. I know they're all still using starfish. I'm not. I know, because I go in the room and I take their starfish thing and I cover it over with this. Okay? So what do we got on this? What do they use on this? By the way, there is the oocyte surrounded by zona pellucida and corona radiata. There's the sperm. So that's not an ovum yet? No, you know what makes that thing become an ovum inside there? When the sperm attacks the corona radiata, that stimulates another division inside the oocyte. Meiotic division, meiosis occurs, and that's what makes the ovum. It's not until it's stimulated by sperm that you actually make it into an ovum. And then the sperm, no, it don't go inside. That's a misconception. All the sperm does is inject, it's 23 single strands of chromosomes inside. The chromosomes enter, but the sperm doesn't. And now those chromosomes join the 23 single strands of chromosomes from the ovum, because now it is an ovum, And then you make 23 pairs of chromosomes, it duplicates 46 pairs of chromosomes. Now you got, boop, B is the zygote. B is the zygote. Next, C is morula. In the human, by the way, this gets up to 64 before it becomes a blastocyst. D here, that's blastocyst. That's the hollow ball with the inner cell mass. The inner cell mass reorganizes, makes the 3 germ layers. There it is, 27, that's 3 layers thick. On top is amniotic cavity. On the bottom is yolk sac. In the middle here, 27, is the embryonic disc. The top layer of cells next to the amniotic cavity is ectoderm. The bottom layer of cells next to the yolk sac is endoderm, and that little rut area in between them is mesoderm. One more thing down here, number 40. This is a late embryo. That is the body. It's not an umbilical cord yet. It's almost there, but this is the body. Allantois. Allantois. Just think of a guy named Alan. I had a friend named Alan, so that was easy for me. Allantois, Allantois, Tois. That's what 40 is there. So I do 4 questions on this. Your professors will do 2, and they'll give you 2 starfish questions. That's it.